AP 120, Chapter 2, the topics proteins and nucleic acids. So we're moving on to the very important macromolecules, proteins. These are very critical to living things, and proteins do a lot of things for our body. So a protein is a long chain of amino acids. Sometimes you'll see proteins called polypeptides because peptide bonds connect amino acids. Poly means there's many of those peptide bonds because there's many amino acids. An amino acid is the monomer for proteins. So a long chain of amino acids, the amino acids themselves are discrete units. They are the monomers. There are three main portions to a amino acid. There is the carboxyl group, which got COOH. There is the amine group, NH2, and then coming off of the central carbon is the side chain or R group. And it's the R group, the side chain, that varies, that is different from one amino acid to another. So this portion here is constant, the same in all amino acids. The R group, the side chain, is different. And here is just a view of alanine, the amino acid alanine. So again, carbon, carbon, nitrogen. Here's the amino group. Here is the carboxyl group. Here's that central carbon. Here is the side chain, which happens to be CH3, methane. All right, so proteins have complex shapes, and those shapes are critical to the functions of the proteins. So we're going to talk about some of the different levels of complexity found in proteins. To start off, you have the primary structure. Primary structure is just the sequence of the amino acids in the chain. What is the order? So a bunch of amino acids get covalently bound to each other, giving us a long chain. If it's a shortish chain, it might be a polypeptide. If it's an extremely long chain, then it is definitely a protein. Proteins tend to be bigger than polypeptides, but to be honest, a lot of sources use polypeptide and protein interchangeably. All right, secondary structure. Secondary structure is when those long chains arrange themselves in either sheets or coils. So secondary structure is either the forming of sheets or coils out of the original long chain of amino acids. And this is influenced by hydrogen bonding between the R groups, between the side chains of the amino acids. That helps to cause this to form. Then there is tertiary structure. Tertiary structure is basically how the sheets and coils interact with each other, it causing the protein to fold and twist up even more, leading to its globular form. So here you can see the sheets and coils have come together to give us this specific tertiary structure. And again, influenced by R groups, the side chains of amino acids. And this can include hydrogen bonding, the attraction or repulsion of charges, and also hydrophobic or hydrophilic interactions. Finally, the fourth level of complexity is the quaternary structure. It's worth to note that quaternary structure is when two or more chains, two or more polypeptides, join into a larger molecule, join together to form the larger protein. Now this occurs in some proteins, but not all proteins. So many proteins have just a single chain of amino acids and go up to tertiary structure, but a few proteins have to have multiple chains come together to form a complete functional protein. So quaternary structure, two or more chains joined into a larger molecule. Again, as I mentioned earlier, the protein shape is very important to its function. Often the shape of a protein is referred to as its conformation. So the protein shape is its conformation. For instance, enzymes, which we will be talking about a lot throughout the semester, have a specific shape that includes a place for a substrate to bind to. And the shape of the protein is appropriate only for that one substrate. So here in this cartoon simplified version, A is going to fit into the protein, B is not. So the protein, the enzyme will only act on A. Sort of how a lock and key works. You need a very specifically shaped key to fit a very specifically shaped lock. Functions of proteins. They can be structural materials to give the cells its shape. They can be an energy source. They can be broken down to make ATP. Certain hormones are based on proteins. Proteins can be receptors on cell membranes, so the cells can be aware of what's going on around them. Proteins can be antibodies to fight infections. And as I already mentioned, proteins can be enzymes that catalyze metabolic reactions. What do catalysts do? They make chemical reactions faster. They speed up the chemical rate, the rate of the reaction. All right, proteins can be denatured. What this means is the protein's shape is disrupted in a permanent manner, making the protein non-functional. So denaturation disrupts the protein's shape. It's a permanent change, and it makes the protein non-functional. So, for example, the enzymes lose their active site, lose the place that the substrate binds. So now it can't bind substrates anymore. Ways you can denature proteins include pH extremes, either super high, super low, depends on the protein, super high temperatures, radiation, chemicals, there's probably some other things out there too. Basic idea being that you can't unfry the egg. Sorry, once you've fried your egg, you can't unfry it. All right, moving on to nucleic acids, the last of the macromolecules important for living things. Nucleic acids are composed of nucleotides. So the monomer for nucleic acids are nucleotides. Here is a cartoon of a nucleotide. Nucleotide has three main components. It has a phosphate group, it has a 5-carbon sugar, and it has a nitrogenous base. So phosphate group, 5-carbon sugar, and nitrogenous base. Turns out there are 5 different nitrogenous bases. So nucleic acids, two big kinds are RNA and DNA, deoxyribonucleic acid. RNA tends to be one single long chain of nucleotides, and its 5-carbon sugar is ribose sugar. So ribose sugar is the sugar for RNA. DNA, on the other hand, is usually 2 strands of nucleotides, so 2 long strands of nucleotides that are covalently bound to each other. And its sugar, its 5-carbon sugar, is deoxyribose sugar. Now, these 2 strands of DNA are connected to each other by hydrogen bonds between the bases of the nucleotides. So that's how the DNA gets its double helix form, 2 strands connected via hydrogen bonding. So RNA functions in protein synthesis. It's important for the production, the making of proteins, also known as translation. RNA can be a messenger RNA, a tRNA, or an rRNA, mRNA, messenger RNA, tRNA, transfer RNA, rRNA, ribosomal RNA. The RNA starts off as a long single strand. Messenger RNA stays one long single strand, while tRNA, on the other hand, ends up forming a complex shape with hydrogen bonding between nucleotides within that one strand. Folds up to be functional. DNA, its big function, as we know, is to store the genetic code in genes. So our DNA, we have something like 20,000 genes. Each individual gene leads to the production of a protein, and it's the genes that determine many of the characteristics of each of us. So the DNA, as I mentioned before, 2 strands forming a twisted double helix. So strand 1, strand 2, strand connected by the hydrogen bonding between the bases, and hydrogen bonding between the bases. And then there is a third nucleic acid called adenosine triphosphate. Adenosine triphosphate, you take a single nucleotide, a single ATP nucleotide, called ATP because the base is adenosine. So A for adenosine, T for triphosphate, P for the phosphate as well. So adenosine triphosphate. Now, this is important because it has 3 phosphate groups. And the bond between the second and third phosphate groups is a high energy bond. And that energy can be used for chemical reactions in cells throughout the body. So ATP is the energy currency of the cell. It is the compound that carries energy that our cells use for many, many chemical reactions. Biomarkers. Biomarkers are chemicals in the body that can potentially indicate a disease or a exposure to a toxin. And sometimes the presence of the biomarker is perfectly fine at some levels, but a problem if it's at other levels. So biomarkers, chemical in the body that can provide information about disease or exposure to a toxin. Now, biomarkers should be easy to obtain and measure. So we want to be able to easily determine if you have the biomarkers indicating a particular disease, right? Sensitivity, it needs to lead to few to no false positives. Only want to say you have something if you actually have it. Specificity, which means that it has little to no false negatives. So if you do have it, you want the biomarker to show that. And good biomarkers work for most everybody. Should be able to use for pretty much anybody. So some examples include cholesterol. Many people have their cholesterol levels tested for, determining if they have too much of the bad cholesterol. Glucose levels often get checked in people, especially people with or thought to have diabetes. Can find chemicals from smoking in a person's body that could be used for some reason, I don't know. Toxins like lead can be detected in the person's bloodstream in their body to let them know if they've been exposed to that. And there are a lot of other biomarkers out there that get tested for. And that is it for this chapter.